All right, welcome back to genetics. And today we're going to be talking about the genetics of bacteria and their viruses. This is all going to be about how bacteria share their DNA. But before we get onto that, we have a little bit of unfinished business from the previous lecture to get to. So we're going to start off with what's called Sanger sequencing. It is the most common type of DNA sequencing, or at least it was until about 5 years ago. And then we're going to get into bacterial genetics, some useful types of mutants, and transformation. But I want to get you started on DNA sequencing first. So this is part of our genomics that we were talking about in the previous lecture, and I cut us off for time. We're going to be talking about Sanger sequencing today. So in the early 1970s, well, mid-1970s, people at that point had been able to identify amino acid sequences from proteins. And they had only just recently discovered that the genetic code was 3 bases long. It was about 15-year-old information at that point. And they had really only discovered most of our understanding of DNA and proteins within the 20 years preceding that. But at that point, there was no way to tell what the sequence of a gene was. They had restriction enzymes barely at that point. I think they had just been discovered a few years prior, and they weren't really in large use. So you couldn't even make restriction maps of DNA. People were really in the dark. And two different labs about the same time developed methods for identifying the specific sequence of bases in a stretch of DNA. And that was a huge game changer. I mean, that was major. The Human Genome Project didn't start until 1988. This is a little over 10 years before that sequencing was invented. And the two different labs that invented it went about it through two different ways. There was the Sanger method, and I believe his first name is Frederick Sanger. And then there was the Max M. Gilbert method. I'm not going to be talking too much about Max M. Gilbert, but I do want to introduce to you Wally Gilbert. He is a delightful man. I've spoken with him a few times. He originally was a physicist working at Harvard in the 1960s. And while he was working there one day, he always had a slight interest in biology, but he was mostly just a physicist. And at this point, Jim Watson, of Watson and Crick fame and infamy, was also working at Harvard. And Watson wandered over to Wally Gilbert's lab one day and said, I need an extra pair of hands for this experiment we're doing. Are you free? And Wally said, sure, why not? I'd love to take part in a biology experiment. So he walked over, and Watson basically said, here, hold this test tube of radioactive phosphorus for us. And he brought Wally Gilbert into his experiment, and the experiment that they were doing was proving the existence of messenger RNA. And so his very first foray into biology was an incredibly major discovery that, honestly, was worth a Nobel Prize. Sidney Brenner and his group working from Pasadena in California at Caltech, they did the brunt of the work, and they had a much more elegant experiment. But between the two groups discovering messenger RNA, there should have been a Nobel Prize. But there wasn't. So Wally was so excited by, hey, my first experiment, and it was really important and major and successful. He was so excited by that he decided to leave physics and become a biochemist. And within 10 years, he had discovered that there was a tremendous need to understand the sequence of bases, and so he set about trying to determine a way to do it. The method he determined was incredibly clunky and difficult and roundabout. It worked well, and it got the job done, but it wasn't clear, simple, and elegant. Sanger, from Sanger sequencing, his method was clear, simple, and elegant. And for this, they shared the Nobel Prize. Wally Gilbert and Sanger and Gilbert's lab partner Maxim. And so we're going to get into the elegant version of DNA sequencing here. And it all boils down to the structure of the sugar. So these are your nucleosides and nucleotides. It's the sugar bound to a base is the nucleoside, and the sugar bound to a base and a phosphate is your nucleotide. We've gone over this before. Now, the phosphate of one nucleotide binds to the 3-carbon of the sugar of the preceding nucleotide, right? We've gone over that. And the 2-carbon in RNA is an OH, and in DNA is just an H. And you can see that 2-carbon is labeled with a number 2. So in Sanger sequencing, you use something called dideoxynucleotides. So what are we talking about here? Well, you know what a deoxynucleotide is because it's deoxyribose. So ribose is the sugar used in RNA, and it has an OH in place of the H at the 2-carbon, like I just said. If you remove the oxygen, you're making it deoxy. So instead of ribose, you now have deoxyribose, and that's the sugar that we're looking at here. You have removed one oxygen from the ribose, and so it is deoxy. Well, in dideoxy sequencing, you're removing a second oxygen, and that is the oxygen on the 3' carbon. So in this case, where you have the OH on the 3, that's just going to be an H for dideoxy sequencing. And that has a tremendous effect because the phosphate of one nucleotide tries to bind to that OH, and it requires that 3' OH to create the bond. And without that O in the OH there, the phosphate cannot bind to the sugar. And so you cannot extend your DNA molecule any further 3' because there's no more phosphates that will come in and bind and become the next 3' base down. Okay? That is the underlying principle of Sanger sequencing. So let's have a look at this. Okay, so here is a DNA chain that's 3 bases long, and as you can see, let's look at the phosphate in between the top base and the middle base. So that phosphate was originally part of the base in the middle, the G, and it bound to the 3' OH and spit off an H2O when it formed that bond. And so now the G has bound to the A. And then along comes the C, and its phosphate binds to the 3' carbon's OH from the guanine. And when it forms that bond, H2O gets spit off, and the phosphate binds to the carbon. And now you've got, on the C, you have a 3' OH. And that's ready and waiting for the next base to be added onto that. Right? I hope none of this is shocking to you. This is how DNA is polymerized and how your strand of DNA elongates from the 5' end to the 3' end. So what happens when we add a dideoxynucleotide? So in every other way, it is identical to all the other nucleotides that we're dealing with. It's still got one of the 4 bases attached to it. It's still got a phosphate attached to it. The only difference is in the sugar, where there should be an OH on the 3' carbon, as you see on the top, there's now just an H on the 3' carbon. The diagrams to the left here illustrate the actual reaction of the phosphate to the OH. It spits off the 2 phosphates that are on the end as a pyrophosphate, and it breaks off an H2O. Okay, so what happens when we add a dideoxynucleotide here? It ends any and all elongation of the strand of DNA. How does that help us? Well, it's funny you should ask. There's a couple of different ways that this can help us. Okay, so first and foremost, let's say that we're doing a polymerization reaction in a test tube, like a PCR, or even just replication. And we add a whole bunch of deoxynucleotides ribonucleotides, so regular DNA nucleotides. When we throw in our polymerase and we put in all the correct ingredients that we need and a primer and so on, it replicates the DNA and creates a whole new double helix, right? We covered all that in the previous 2 lectures. Between the PCR and the DNA replication, you understand how that works. Now, what happens if we add mostly deoxynucleotides, but a small proportion, maybe 20 to 30%, dideoxynucleotides? Well, so here's what's going to happen. Randomly, the polymerase is going to grab a nucleotide. If it grabs a deoxynucleotide, a regular DNA nucleotide, it's going to grab that, add it onto the DNA chain, and move to the next base and grab another one. And if it grabs a regular deoxynucleotide, it's going to grab that, it's going to add it on, and it's going to move on to the next one. Now let's say it grabs a dideoxynucleotide. Grabs that, adds it on, it cannot add any more, so it leaves. And so now we have only polymerized 3 bases. But there's a whole lot of single-stranded DNA in this test tube, so the polymerase binds to a primer on another single strand of DNA, and it adds a base, and 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 then a dideoxy base. And so that's added, 5 or 6. And then it leaves, and it goes on to a different single-stranded DNA with a primer. And it adds 1 base, but that base was a dideoxy, so the very first base it adds, it falls off because it can't add any more. You're going to get a whole lot of strands of different lengths. Randomly, completely randomly. And if you run those strands on an electrophoresis gel, what you're going to get is a large number of bands showing different DNA fragment sizes. And the fragment sizes are basically going to be 1 base length apart, because randomly you'll have a bunch that have only been extended 1 base. You'll have a bunch that have been extended 2 bases, a bunch that have been extended 3 bases, all the way up to the length of your full DNA. Okay, great. How does that help us understand the sequence of the DNA? Well, what if I told you the dideoxynucleotide could have something else attached to it? This is easy to do. We can find out ways to attach things to the nucleotides that will not get in the way of the polymerase, and so they will be treated like a normal base. And if that happens to be fluorescent, then we will know that a base was added there when it shines fluorescent. And if we put a base that shines fluorescent green for a G, and fluorescent purple for a C, and fluorescent red for a T, and so on, then depending on what color fluoresces, we know which base has been added. Okay, that's great. But I don't want to sit there and stare at a gel under UV light and count green, red, orange, red, purple, black, whatever. That, especially if you've got like a 4,000 base long gene, you don't want to do that. You're going to make mistakes, and your mind's going to go numb, and your eyes are going to water, and it's a bad situation. So what Sanger further developed, in addition to all of this, was, originally speaking, instead of adding a fluorescent tag to the bases, the dideoxy bases that he added had radioactive phosphorus in the phosphate. And that meant that, if he added normal nucleotides that were not radioactive, and then only the dideoxynucleotides were radioactive, then when you did this polymerase reaction, the very last nucleotide of any given fragment of DNA, the 3' most nucleotide, would be radioactive. So you set up 4 test tubes side by side. And in the first test tube, you only add radioactive adenine. You put in mostly regular nucleotides, and you put in radioactive dideoxy adenine. Okay. And then the second test tube, You add all the regular nucleotides, and then you add radioactive dideoxycysteine. In the third test tube, guanine, radioactive dideoxy, and in the fourth, thymine. Why? Well, in the first test tube, your polymerase is going to be going along, it's going to add bases here and there. Any C, G, or T that it adds is going to be normal. And, 80% of the adenines it adds are going to be normal. And then every once in a while it's going to throw in an adenine that's dideoxy, and it's going to stop. But it's only ever going to stop on the adenines because those are the only ones that are dideoxy. But every dideoxyadenine is radioactive. So in that test tube, you will have a bunch of DNA fragments of different sizes, and the last base of each size will be an adenine that is radioactive. In the next test tube over, it's the same thing, but the last base of each fragment there is a radioactive cysteine. Now, the fragments are all going to be different sizes because the A's and the C's are in different places. I'm going to demonstrate this in the next slide. So look on the right side of the screen. And what you're looking at there is a cartoon of a gel showing what you would see. So you run this gel with, in the first lane, one of the 4 radioactive bases, in the second lane, the next radioactive base, and so on. You've done the polymerase reactions, the reactions are done, and now you add them to your gel and you run the gel. Well, you can't see the radioactivity with your naked eye, but you know that when the radioactivity gives off photons of energy, that would make film be exposed. Now, I know you guys are raised in the era of digital cameras, but bear with me, there's this thing called film that people used to take photographs on, and exposing it to light would make it turn darker. And depending on what kind of chemicals you use to process it, you could actually see the colors. Well, in this case, you put the film up against your gel, and you put it in the freezer for a day or two, and when you take it out, you develop the film by putting in a couple of chemical baths, and wherever there was radioactivity touching the film, it exposed it to a little bit of light, and that light made the film turn black. And so what this image shows is a cartoon of a film that was exposed to this gel. And if you look at the bottom of the gel, that's where your smallest fragments are. And at the top of the gel is where your longest fragments are, because that's how gels run. The smallest fragments go further faster. We covered that in, I think, lecture 2 even. So if you want to find out what the sequence of your DNA that you were just polymerizing is, you look at the very bottom band and you see which lane it's in. That's in the lane for A. So your very first base in your DNA sequence is an A. The second base is the band directly above that in whatever lane. And so you look, and the next base up, the next band up is in the T lane, so your next base is a T. And then you go up from there, and the next 6 bases, or 7, 6, the next 6 are all A's. And then you see a band in the C column, and then a band in the T, and a band in the C, and the A, and the G. And so that's how you read a DNA sequence through Sanger sequencing. It's really simple. You set up a polymerase chain reaction, a PCR, and you do it in quadruplicate, and in one of each, you put in a specific radioactive dideoxynucleotide. You run your gel, and you expose the gel to film, and then you develop the film. It's a lot simpler than the Sanger method, which involved a whole lot of chemical treatments of DNAs with varying concentrations for varying times, and then you would also run it on a gel with radioactivity, but the banding was much more complicated, and you had to add and subtract. You could get the right sequence, but it was a logic puzzle. This is much more straightforward, and you can just read it. So that was the standard of DNA sequencing from the late 70s up until, I'd say, the late 90s, even the early 2000s. And around that time is when people really started getting into fluorescence. in terms of how to use fluorescence. And so we go back to the nucleotides that I was talking about on the previous slide, where each base can fluoresce a different color. And that's what we see here. If we were to amplify the same sequence using the new fluorescent Sanger sequencing, then you would get a gel, but instead of exposing this to film, what actually happens is the gel apparatus itself is connected to a machine with an optical scanner. And the optical scanner reads fluorescent wavelengths. And you turn on the gel, you run the electric current through the gel, and the DNA migrates. And as each band of DNA migrates past this optical scanner, it can read the fluorescence. So you are getting real-time results as the gel runs, as each band of DNA moves past it. So each fragment of DNA, with its one fluorescent terminal nucleotide, will be seen. And in this case, you can do it all in one test tube. You don't have to do it in separate test tubes, because instead of just having a black band on a black and white gel, where you need to distinguish them left to right, because they all look identical, in this case, because each one's a different color, you know for sure that band is a T, even though it's directly on top of the A band. I mean, if you look at the black and white gel, if those were all in one column, how would you tell which one was which? You couldn't. But because with the fluorescence they're all different colors, then you can tell. And so for this, instead of reading which column each band is in, you can just look at the colors of the peaks. And those peaks represent intensity of light at a given color wavelength. And that's how you read Sanger sequencing DNA. And that was the standard from late 90s, early 2000s, up until about 5 years ago. That is still the standard for performing small DNA sequencing reactions. And in a sense, that is also the standard for next-gen sequencing. So next-gen sequencing stands for next generation. And instead of doing one DNA sample at a time in a test tube, you can do literally thousands of different DNA samples in the same test tube. And the machine that reads the fluorescence can distinguish between them. Because if you think back to what I said about PCR, how you can add random sequence to the 5' end of your primers, and it won't get in the way, and it'll be added onto your eventual replicated DNA. The way next-gen sequencing works in many cases, and there's a handful of different ways that different companies do this, but they all more or less boil down to this. They create a specific DNA sequence that's kind of like a barcode. They create 1,000 of them, and each one's specifically different from the others. And that's, 20 bases long, let's say. And, if you think 20 bases, a 20 base long oligo can have any one of 4 to the 20th sequences. So that's billions. So you don't have to worry about repeating this specific sequence, these barcodes. So they create these barcode sequences and then random primers of 10 to 20 bases long attached to them. And then they perform in one test tube thousands of different amplifications. So you have genomic DNA in there. You've got thousands of different random primers that each individual random primer sequence has its own barcode. And you perform your amplification reaction. And what you also have is a chip, like a microscope chip. And fused to that, you can actually bind DNA to chips. So fused to that, you have the complementary DNA sequence to each of the barcodes laid out in a grid. And you can basically perform the polymerase chain reaction with the chip in the test tube. Well, it's not really in a test tube. You've got a chip underneath an optical scanner, and the PCR is done directly underneath the optical scanner. And every time a base gets added on to the chain of nucleotides, it fluoresces. So what you have is a grid of specific points of DNA, and each one of these grid points fluoresces intermittently as a new base gets added on. And the optical scanner reads all of this, and instead of a vertical line of peaks of fluorescence that you can read base, 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 instead it is a time sequence of, at 1 second, these are the 1,000 different fluorescing colors, one at each of these 1,000 points on this grid. And then 1 second later, here is all of the fluorescing colors, and each one's now different. And by reading the time sequence that way, the computer can read each of the thousands of strands of DNA as they are being elongated. That's one type of next generation sequencing. And there's a lot of similar types of different methods. So next-gen sequencing can do entire genomes in an afternoon. The hard part is just then the computer software to actually line them all up to make a genome. Because what you have is thousands of smaller sections of DNA that you then have to line up and make sure that they're lined up properly. But that's a whole other can of worms we're not going to worry about. You don't have to worry too much about the specifics of next-gen sequencing and how it works, but I just want to give you an idea of the technology that people have developed just based on how polymerases work, basically, and the chemical structure of nucleotides. I mean, we are manipulating the cellular machinery to do our bidding at an alarmingly rapid pace. It's really cool. Okay, let's talk about bacterial genetics, the actual purpose of this lecture. Now, most of the first half of Chapter 7 of the book, and that's what we're covering today, is the first half of Chapter 7. Most of this is just different ways that bacterial DNAs can combine with each other. And it's kind of redundant because they all more or less combine the same way, but they've got different names for the different things. So let's try and clear this up a little. We'll start with plasmids. So hopefully at this point you all have a general idea of what a plasmid is, because the lab uses them extensively and talks about them extensively. But the way that I like to think of a plasmid is it is an upgrade to a bacterial cell. So bacteria has circular chromosomal DNA, and that has everything that the bacteria needs to survive. It's got, all of the genes that encode for the proteins for replication. It's got the genes that encode for the proteins for metabolism, for cellular processing, all kinds of other things. It's got everything the bacteria needs to survive. It's all in one circular DNA chromosome, and it's, 50 kb, 100 kb long, and that's all the bacteria needs. But there's other circular DNA that a bacteria could have, or could be without, and it just adds functionality to what the bacteria can do. So I think of them as upgrades or apps or add-ons. If you're looking at it as a video game, it's just an equipped tool or an equipped weapon. It just gives you the opportunity to do something new. Most plasmids, what it actually gives is resistance to an antibiotic. What are antibiotics? An antibiotic is merely just some sort of chemical, naturally occurring in most cases, that kills a bacterium. So some bacteria have evolved the ability to make a gene that prevents them from dying in the presence of that chemical, whether it's something that degrades the chemical or prevents it from entering the cell or what have you. So most plasmids are just a vehicle for bacterial antibiotic resistance. Now, the plasmids have their own origin of replication, at least one. Some of them have multiples. And so they amplify, or they replicate, at the same time the chromosomal DNA does. When the bacterium has all of its polymerase and everything like that ready to go, and it duplicates the chromosomal DNA, well, the polymerase can't tell the difference between the chromosomal and the plasmid DNAs. It's just a circular DNA. So the polymerase replicates everything. And so the plasmid gets duplicated and then it migrates into the daughter cells. So once a bacterium gets a plasmid, it pretty much always has it, more or less. But the cool thing about plasmids is they can export themselves into other bacteria. And that doesn't mean they pack up their bags and go. It means they duplicate themselves into the other bacterium. So this slide is badly out of order. I don't know why. It is an image of an agar plate. So these are the kinds of petri dishes that scientists grow bacteria in. And the sort of yellowish blue color in the plate is a jello-like substance that basically contains everything the bacteria needs to survive. And the white circles on there are colonies of bacteria. So each individual one of those circles started off as one bacterium that landed on this dish somehow, and then started growing and dividing and growing and dividing. So each one of those is thousands of copies of essentially identical bacteria. Now, each individual colony could be drastically different from every other colony, but all of the bacteria within one colony are basically identical because they all are daughter cells of one original cell, however many generations later it's been. And I'm not sure why this slide is here. I meant to move that. So I said that the plasmids can export themselves into other bacterium. This process, sort of broadly throughout biology, is called horizontal gene transfer. What do we mean by that? Well, let's think about normal gene transfer. Normal gene transfer is you are person 2-1 here. 2 because you're in the second row, 1 because you're the first person in the row. When you have children, you are passing your genes on to them. I mean, that is the entire purpose of existence from a genetic standpoint. It's to pass your genes on to another generation, to more organisms, so that the genes themselves perpetuate. So how does that work? You are sending your genes, in the parlance of this pedigree, this family tree, you are sending them vertically down to the next generation. That is vertical gene transfer. That is what we are predominantly talking about when we talk about genetics. But bacteria and some other organisms that we won't get into, but predominantly bacteria and viruses, they have vertical gene transfer just like that. I mean, they split into daughter cells through mitosis, and they pass their genes on that way as normal, but they can also do horizontal gene transfer. So let's say that your organism 2-4 in this case. You can actually send your DNA not just down to 3-5 through 3-12, you can send your DNA to organism 2-6, horizontally through the pedigree. It doesn't have to go through meiosis. It doesn't have to go to your offspring. You can send DNA to another organism that is not a daughter cell or a gamete. That is horizontal gene transfer. So how does that work? Well, let's talk about F factors. F factor is a plasmid. Okay, the F stands for fertility because before any of this was understood, it got figured out that bacteria that have this particular plasmid could do specific things having to do with fertility. In that they can transfer this plasmid from one cell to another. So, another word for some kinds of plasmids is an episome. Epi in genetics kind of means on the periphery, on the side, not central. So an episome is sort of a peripheral chromosome. So in this case, it means, as it says here, a genetic element that can insert into a chromosome or replicate as a circular plasmid. All you need to know is it has DNA that gets replicated. Okay? And that DNA contains genes that form proteins that do things. So, one thing that I didn't mention earlier is that depending on how many origins of replication a given plasmid has, it can be quantified as low copy number or high copy number. Because if it has multiple origins of replication, it winds up making more copies. And if it has fewer origins of replication, it winds up making fewer copies. So the F plasmid exists as really only 1 or 2 copies per cell. A high copy number plasmid could be anywhere from like 10 to 50 copies of the identical plasmid in a given bacterium cell. So the F plasmid is pretty big. It's as big as most bacterial chromosomes because it encodes a lot of proteins. And as such, it is a low copy number plasmid because it's bulky. And as a typical plasmid, it replicates during the cell cycle and segregates to both daughter cells. So what does it do? Why are we talking about F factor? Well, let's get into that. F factor initiates something called conjugation. And yes, that is the same usage of the term conjugation as sexual conjugation or congress. This is an exchange of DNA between two organisms. Now, the F plasmid encodes for a bunch of proteins that form little finger-like projections sticking out of the bacterial cell membrane. These are called pili. An individual is called a pilus. And they just stick out from the cell membrane a little ways, and if it bumps into another bacterial cell, then it latches onto that cell and forms a tube connecting the cytoplasm of both bacteria. And so now these are two independent bacteria, but they are now connected. And their cytoplasm can migrate freely and intermingle between the two of them. And what happens next is the plasmid undergoes DNA replication and spits out one strand of that DNA through the tube, this pilus, connecting the cells. So, if you remember back to my original lecture on mitosis, and I was talking about the microtubules that come out from the spindle, and they find the kinetochores and bind them, and I gave that analogy of pulling on a rope, and the rope is constantly getting shorter from where you're pulling on it, and it's not getting any longer on the other end anymore, and so as you pull and it gets destroyed behind you, it gets shorter and shorter, and you pull things closer and closer to yourself. Well, that happens also here with the pilus. The pilus reaches out from the cell that has the F plasmid, and we're going to call a cell that has the F plasmid F+, and we're going to call a cell that does not have the F plasmid F-. So the F+ cell forms a pilus with the F- cell, and then it starts shrinking the pilus from its own side and bringing the F- cell closer and closer until the pilus between them is very short and the cells are very close together. That's what we're seeing on the left side here. On the right side, we're not showing the bacterial chromosome at all. We're just showing the F plasmid. That is going to undergo rolling circle replication. Now, if you remember rolling circle replication, I mentioned that briefly as one of the possible methods of replication during that lecture. And it's basically one strand of the DNA gets nicked, and then it unwinds, and as it's unwinding, the polymerase just hangs out on the remaining circular strand and synthesizes DNA as the strand spins in a circle through it. And the other end is spit out, and there's Okazaki fragments being generated on that. In this case, rolling circle replication occurs, and the leading strand, the continuous strand of replicating, polymerizing DNA, is the part that is inside the F positive cell. The lagging strand gets shunted through the pilus into the F minus cell, and the F minus cell synthesizes the Okazaki fragments and then ligates them together. And so what you have at the end of that, the pilus disappears between them, and now you have a copy of the F plasmid in what was originally an F minus cell, and now you have two F plus cells. So that is, you're just making a copy of the plasmid through DNA replication. That's all you're doing. It's just that one strand is being synthesized in this cell, and one strand is being synthesized in that cell. And what you're looking at is semi-conservative DNA replication across two different cells. So it's kind of like normal mitosis in that respect, except you start off with two cells, not one, and only one of them has the DNA you're talking about to begin with. So F plasmid is transferred through rolling circle replication in a pilus. And that's what we're talking about here. That is one way that bacterial DNA can get from one cell to another. And in fact, it's a main way, because we're going to talk about a couple of other things now. We're going to get back to this transfer in a few minutes. But I want to talk to you next about transposons. Transposons I also mentioned in a previous lecture, and I didn't get into them in detail. I just said that they're stretches of DNA that can hop around. They can cut themselves out and go to a different place. They're called jumping genes. They were originally discovered in corn by Barbara McClintock at Cold Spring Harbor. I mentioned Cold Spring Harbor a lot. A lot of these discoveries were made there. So what is a transposon? Well, a transposon is... See, a lot of entities in molecular biology are referred to with the suffix of on, or son, zon. You know, you've got introns, exons, cistrons, transposons, integrons. There's a whole bunch of different things, and that just means a unit or an entity. And in this case, a transposon is a unit or entity that transposes itself. It moves from one place to another. So what is a transposon? Well, a transposon is a stretch of DNA that encodes a protein called transposase. And the ends of that stretch of DNA are, just like most restriction binding sites or restriction recognition sequences, they are palindromes. So the end of one side of the DNA is the inverted repeat of the end of the other side of the DNA. The whole thing, all the way through, is not a palindrome, just the ends. And I want to be clear here, it's not that the left side is a palindrome of the left side, and the right side is a palindrome of the left side, of the right side, rather. So it's not like a restriction site in that sense. It's more that the left side is the inverse repeat of the right side. And so just the ends form palindromes of each other. I want to be clear on that. Now, the sequence of DNA between these inverted repeats can include a lot of things, but it always includes a gene encoding transposase. And the gene encoding the transposase, so you see the transposase there as a red squiggly line, and that's supposed to illustrate the protein of the transposase, or maybe the RNA, I'm not sure. But we're going to get into the specifics of translation later in the semester. But hopefully you're a little bit familiar with it from Bio 2000 or 1000, in that the RNA gets read by ribosomes to create a protein sequence through a chain of amino acids. And the RNA, the first codon in the RNA that gets read is for a methionine, an ATG base sequence that encodes a methionine amino acid. And the ribosome starts there and creates the amino acid polypeptide from there. Well, the transposon, the gene for the transposon, has multiple methionines in it. I mean, almost all gene sequences do. But the ribosome in this case starts at two different methionines. And if it starts at the earlier methionine, it creates a full-length sequence of amino acids that forms transposase. But if it starts at a specifically later methionine, it creates a shorter version of the transposase that is called a repressor. And it's called a repressor because it represses the activity of the transposase. It gets in the way and stops it from working. It gums up the works. Because it's mostly the same as the transposase, but the part at the beginning is kind of important for function, and when the repressor gets in the way, it doesn't have the ability to function the way the transposase does. So it blocks the transposase from working. And so what you have is, frequently, throughout the life of whatever organism, the ribosome is only starting at the repressor part. And so nothing happens. But every once in a while, the ribosome will start at the true beginning, where the transposase gene starts. It'll start that part of the mRNA and make the full-length protein sequence, and then you have a functioning transposase. And what does the transposase do, you ask? The transposase cuts the inverted repeats like a restriction enzyme. And it cuts out the insertion sequence, the actual transposon, and it moves it. Now, that's the top half. The bottom half of the image here, it has two of these insertion sequence things. So the insertion sequence is the stretch of DNA with the transposase gene and the two inverted repeat ends. Many transposons are actually tandem. They have two insertion sequences with a stretch of DNA in between. And of the two insertion sequences they have, one of them has a bunch of mutations that prevents it from making transposase or repressor. And so only one of them actually makes the transposase and repressor. And that's the lower image. You can see that there's only the red squiggle and the blue squiggle on the right side, not on the left. Because in this case, the left side has mutations. But it also has insertions of a bunch of, in this case, antibiotic resistance genes. This one in particular, the neo is resistance against neomycin, the ble is resistance against bleomycin, and the str is resistance against streptomycin. So streptomycin is called that because it's useful against streptococcus, which gives strep throat. So if you have If you are infected with a bacteria that has streptomycin resistance, then you can't take streptomycin to kill your bacteria, and the strep throat stays until you get something else. So in this case, these antibiotic resistance genes have become inserted between the insertion sequences here. And now when this transposon gets activated, it's going to cut the far left and the far right, and that whole long stretch is going to get taken out of whatever plasmid it's in or bacterial chromosome, and it's just going to up and pack its bags and move somewhere else and plop down and insert itself into a chromosome or a plasmid somewhere else. Just going to get up and migrate to a new location, and it's going to be a squatter there. That's transposons. That's how they work. They cut at the inverted repeats, and they move the whole insertion sequence with them. And why did they take the insertion sequence with them? Because that's the DNA that encodes the transposase. They need that. If they don't take it with them, they are no longer a functional transposon. And once they move to the new place, if they obtain too many mutations through DNA replication, they will no longer have a functional transposase. They will no longer be a functional transposon. They will be a ghost of a transposon. They will be DNA baggage. You would be shocked to learn how much of the human genome is non-functional transposons. Shocked, I tell you. Let's talk about co-integrates. Let's say that you have a transposon, and the transposon has inserted itself into a plasmid. And at some point, the transposon jumps out of there, and it inserts himself into the bacterial chromosome. Now, great. It's no longer in the plasmid, and now it's in the chromosome. But there was more than one copy of that plasmid in the cell. An identical copy of the plasmid. And now, the chromosome and the plasmid both have this transposon identical in each circular DNA. And just like in meiosis or mitosis, when you're lining up chromosomes next to each other that have identical sequences, they can start forming chiasmata and homologously recombining. The plasmid and the bacterial chromosome do the same. They overlap, they connect, they recombine, and now what you have is the bacterial chromosome with the plasmid DNA sequence in it. I hope you can see clearly from this visual how that happened. So this is called co-integration because co meaning the two of them and integrating, you're combining them together. And so the thing at the end, the bottom image, that is circular chromosomal DNA, but it is also called a co-integrate because it has the plasmid DNA within it as a linear section of the circular chromosomal DNA. Now, this process is reversible. Think about that, because it's still a sequence of DNA flanked by identical sequence. So at some point, it can homologously recombine again and kick itself out. And it's really just random chance, just like it's random chance for homologous recombination during mitosis of sister chromatids or homologous chromosomes. The same thing here. It's random chance whether those stretches of DNA line up next to each other, because you have to remember, in the cell, the circular chromosome does not look like a circle. I mean, it's not laid out flat in a circle. It's all just sort of wrinkled and bundled up and folded on itself. It's packed in. It's not laid out for display. It's jumbled and packed in. And in that packing, different sections of DNA could be next to each other. And if the identical matching base sequences of DNA happen to line up next to each other, there's a good chance that they're going to cross over. and you can spit the plasmid back out. Now, let's say you have a small plasmid that inserts itself into the F plasmid. And then the F plasmid forms these pili that then bind to another cell, and the F plasmid undergoes rolling circle replication. And now the new F plasmid is in this other cell, but the smaller plasmid had inserted itself into the F plasmid before that happened. And so now there's a copy of that smaller plasmid DNA in the F plasmid in the second cell now, and then it kicks itself back out. That plasmid hitched a ride and took advantage of the F plasmid's ability to horizontally gene transfer, and it transferred itself to another cell. Now, I'm not saying this was a conscious, deliberate decision. That's not how this works. You're just looking at an inert chemical. But the effect is the plasmid got horizontally transferred. This is the underlying cause of antibiotic resistance in medicine. This is why several antibiotics no longer work. Because what happens is, if you get infected with a bacteria and you take antibiotics for it, if you don't kill all of that bacteria, it's because some of it had an antibiotic resistance gene. Maybe you got infected with a non-homogeneous population where some of them had the plasmid, some of them didn't. Those bacteria that are in your system are going to undergo gene transfer to extend the antibiotic resistance gene throughout the population. Because as long as you're taking the antibiotic, the ones without it die, and the ones with it survive. If you stop taking antibiotics too early, then what you are basically doing is you are leaving enough bacteria in your system that they can repopulate. If you take the if you take the antibiotic for the full extent, you are drastically reducing the number of bacteria to the point where your body can deal with them on its own and it doesn't need external help to kill them. So why is it bad to stop taking the bacteria sooner? Well, first and foremost, you're still sick, you're still infected, but also now you have selected for a population of bacteria that have resistance to this antibiotic. And they can pass that plasmid that carries the antibiotic resistance gene to any and all bacteria in your system. And humans are shedding bacteria constantly. We actually are 10% human cells, 90% other cells, including bacterial cells, viral cells, nematodes, all kinds of things. Our bodies are less than 10% human. And so you are now spreading this antibiotic resistance gene through all of the bacteria in your body and in the cloud of bacteria that surround us. Our bodies actually extend a good 3 or 4 feet off of our skin as a cloud of aerosolized bacteria that we are constantly shedding. And when we get in close contact with other people, a lot of those bacteria can overlap or become part of the cloud of the other person and get taken up internally into their bodies. And now they have bacteria with a plasmid that has antibiotic resistance, and that gets horizontally gene transferred into them, and so on. And if they already had a strep-resistant antibiotic plasmid, and they also now add on this ampicillin resistance or bleomycin or neomycin resistance or whatever, they're now building up bacterium that are resistant to multiple antibiotics. And those eventually get passed throughout the population until most of the bacteria that we pass and forth pass back and forth amongst each other to get each other sick all the time, they start accumulating over and over and over again multiple different antibiotic resistance genes. And the antibiotics then are useless. So the reason that we need to make sure we do not stop taking antibiotics early is so that our body can successfully kill off the antibiotic-resistant bacteria that we have without needing antibiotics, so that we aren't propagating the plasmids with antibiotic resistance throughout the population. Okay, I'll get off my little soapbox now and let's move on. What we were looking at here is between the co-integration and the transposons, we're just looking at ways that DNA can get broken and move around, right? It gets cut and it moves around to a different spot of DNA and inserts itself, or there's homologous recombination and the DNA moves that way. Well, there's another version of this that it's more or less the same, and this is called recombinases. And recombinases are just enzymes that stimulate recombination. Homologous recombination can happen on its own, or it can be nudged along by a recombinase enzyme. But the difference with recombinases are it's not just any stretch of homologous DNA side by side. It is specific recognition sequences of DNA. So in this example, we're looking at how Cre recombinase works. Cre is the name of the enzyme, Cre recombinase, and the sequence it recognizes is called a LOXP site. LOX capital P. The LOXP site is 34 bases long. And if there are two stretches of DNA with LOXP sites near each other, Cre recombinase will come along, and if it sees them, it will initiate a holiday junction, which you might remember from earlier discussions of crossovers. And the holiday junctions, thanks to the recombinase, the holiday junctions get resolved by switching strands of DNA. Now, if you pay attention to the colors of the DNA strands here, the light blue, dark blue, light red, dark red, you can see at the top, one double helix is all blue and one double helix is all red. And after the resolving of the holiday junction, you look down at the bottom, and the blues and the reds have migrated somewhat, and they're no longer together. Or if you look carefully, on the top strand, the two blues are still together on the left, and the two reds are together on the right. And on the bottom strand, the two reds are together on the right, and the two blues are on the left. So in this case, if you're looking at this blue plasmid and the red, what looks to be a linear segment of DNA, then on the bottom of this image, what is not shown is the blue on the left of the top double strand of DNA goes off to the left and then loops around to the right and connects to the bottom strand of DNA there. And that is how they are now connected. I know that's confusing. So let's look at it this way. The Cre recombinase has just introduced this kind of insertion into the red or pink DNA. This is what we saw with the homologous recombination in the previous slide, right? Well, so now it is just being done by an enzyme. It's the same process, but it's being done by an enzyme. The stretches of those double helices that are one strand red, one strand blue, those are within the 34 base pair LOXP sequence. So they are still identical in sequence, it's just one strand had been on the other strand. This Cre-Lox system, as it's called, because it uses the Cre recombinase and the LOXP sites, this is incredibly widely used in mouse genetics and medical research. And just to point one thing out, the LOXP site is 34 bases long. What are the odds of getting that specific 34 base sequence? Well, it's not just any 34 base sequence, it is a specific 34 base sequence. Well, that's 4 to the 34th power, which is about 3 times 10 to the 20th. So this 34 base sequence would only occur once every 3 times 10 to the 20th at random. The human genome is only 3 times 10 to the 9th. So the chances of encountering a LOXP site randomly in the human genome is almost nil. It's 1 in 100 billion. Okay? So I don't believe there are any LOXP sites natively in the human genome. It's a bacterial thing. However, people have been able to engineer mice to insert specific DNA of our choice into them. And we use the Cre-Lox system frequently for this. So here's an example. Instead of putting the LOXP sites on different chromosomes or on different double strands of DNA, what we can do is we can put a LOXP site on either end of a gene on a single strand of DNA. The Cre recombinase here is illustrated by a pair of scissors in a circle. So the Cre recombinase sees these two LOXP sites, forms a holiday junction between them, and then recombines them. And what winds up happening is the stretch of DNA between the two LOXP sites winds up being kicked out as a circular DNA, kind of like a plasmid, but it doesn't have the things a plasmid needs, so it's just a circular stretch of DNA. And the LOXP sites remaining, the half sites that remain, get stuck together. Now, if you try and visualize this or draw it out on a piece of paper, you will see that this process is exactly the same as what was illustrated in detail on the previous slide. It's just the result is different. So instead of inserting a plasmid into the DNA, we're essentially doing the reverse, and we're extracting circular DNA from linear. So we can do this a bunch of different ways. Now, not only can you delete DNA between LOXP sites, but because of the way LOXP works, if you have the LOXP sites going anti-parallel to each other, facing towards each other, then when the Cre recombinase creates the holiday junction between them, the end result is the DNA in between gets reversed. So you can make a gene go backwards, or you can make a part of a gene go backwards, and that's a great way to make the gene no longer work. Or you can treat it like two different strands of DNA, and you can cause a translocation. And so this is a great way of trying to mimic human genetic disorders in mice. You look at the human DNA of, for instance, a disorder resulting from a translocation, like DiGeorge syndrome. You look at where the human DNA sequence gets broken and translocated, and you introduce LOXP sites into those specific spots in the mouse genome, one on each chromosome, and then you force the mouse to undergo recombination by Cre. And if you engineer it so that this happens in the developing embryo, the end result is you will have pups born with the mouse version of the human mutation. But that's not the most common way the Cre-Lox system is used in mice. I'm going to talk about the LAC-Z reporter system. So I'm going to get into LAC-Z more in Chapter 9. Right now I'll just tell you it is a way to visualize specific regions of DNA. And in this case, what would normally be the LAC-Z gene is replaced in this image by a GFP gene. So if you know what a promoter is the place on the DNA where you're telling the cellular machinery to start making RNA from here forward. And we are going to insert into this region of DNA, between the promoter and the GFP gene, we're going to insert 2 LOXP sites with stop codons in between them. So stop codons are the 3-base sequence of DNA that tells the cellular machinery to stop making RNA, right? Or at least on the RNA, they tell it to stop making protein, I should say. So we're going to make this DNA, and we're going to put it in a mouse. Great, yay, woohoo. Now, if we add Cre recombinase to this, what happens? Well, the Cre recombinase is going to cut the one LOXP site, and it's going to cut the other LOXP site, and it's going to make that the stop with the 2 half LOXP sites into a circular plasmid, and it's going to recombine the other half LOXP sites together. So we're getting rid of the stop, and we're removing that section, and then we're going to close the area between them. And so now, when the cell makes RNA from this, the RNA is going to be able to make that GFP protein. Whereas before the Cre recombinase, when it made RNA from that, any protein that would get made would stop before it even reached the GFP. And so this is a way of saying that wherever the Cre recombinase is, we are going to see GFP. It's going to be a reporter. And wherever there is not GFP, that means there was no Cre recombinase there. How does that help? You'd be amazed. This is some images of a developing mouse. The top is what's called a whole mount image, and the bottom is cross-section images. And so it's quite literally, the bottom is a slice horizontally through the embryo up above it. These aren't identical embryos. They aren't from the exact embryo, but from one that's similar. So in this case, what you're looking at is the result of Cre-Lox staining. What we have is we have a mouse that has LOXP with a stop codon upstream of a gene that makes for it makes an enzyme called LAC-Z. And the short version is wherever LAC-Z functions, you get blue dye showing up. And I'll get into that more in Chapter 9, like I said. But so wherever the blue dye is showing up here, that means that the stop codon had been removed by Cre recombinase, and so now it's showing the LAC-Z. And wherever you don't get the blue, that's because there was no Cre recombinase there. Well, how did we get the blue in these specific spots? And let's talk about what these spots are. Let's have a look at what we're looking at here. This circle shape is called the otic vesicle, and that is going to develop into the ear. Everything above this is going to develop into the brain. Now, it may look like this is going to be an eye, or maybe this is going to be the eye, but the eye hasn't started developing yet. So this is fairly early on in mouse development. The E6.5, E7.5, E8.5, those refer to embryonic day of development. In a laboratory setting, mice are always assumed to be, well, the egg is always assumed to be fertilized at midnight. So if you look at the development at noon of the next day, that's a half a day. And so this is 6.5 days after fertilization. Now, over here, if you look at this sort of pink structure, this is the neural tube. Your spinal column is going to develop from this. Over here, this is the heart. We're looking at 7.5 days. The heart is already beating. There isn't much blood, but the heart is beating at this point. And at 8.5 days, if you look here, this darkening shape in a sort of pointed oval, that's the developing forelimb bud. So that will become arm. And you can just barely see down here at the bottom is a developing hind limb bud. That's going to be the feet. And then from here on down like that, that's all going to be tail. So that's what we're looking at. This is the stage of mouse we're looking at here. The face is actually going to develop from these structures here called the pharyngeal arches. And these are all aspects of development that I would love to get into if I am able to teach a developmental biology class, which hopefully will be occurring next semester or next spring, worst case scenario. So any of you who are sophomores or juniors and you're interested in developmental biology, please keep your eyes peeled for that class. One thing I want to point out is this little blue circle underneath the neural tube, labeled NC. That is the notochord. And we're going to be doing a lab at some point later this semester where the notochord is important. So just keep in mind that that is a structure just beneath the neural tube, and it runs pretty much the entire length. In fact, I'm fairly certain that all this blue line here is the notochord. So it runs the entire length of the long axis of the mouse. So what are we looking at? What is the blue actually showing us? Well, the blue is corresponding with a region of gene expression here. In this case, the gene we're talking about is FOXA2. And the scientists who developed this mouse and wrote this paper, they basically, on one of the mouse's 2 homologous chromosomes, where FOXA2 is located, they replaced the FOXA2 gene with the gene for Cre recombinase. So from one homologous chromosome, the mouse is producing the FOXA2 protein off of its gene, and from the other homologous chromosome, it is producing Cre recombinase instead. And so this shows you where in the mouse and at what different times FOXA2 is being expressed. So the Cre recombinase is there, but then the mouse is also expressing the LOXP sites surrounding that stop codon that I talked about on the previous slide. And that's being expressed throughout the entirety of the mouse. So each one of these lower rows is a cross-section of the mouse above it. So basically, this one cuts right through there, more or less. And this one cuts right through there. The entire mouse is expressing the LOXP in front of the reporter gene. Now, on the previous slide, the reporter was GFP. On this slide, the reporter is LAC-Z, and LAC-Z produces a blue dye when you treat it. In the entire mouse, you have this LAC-Z. I'm sorry. In the entire mouse, you have the Cre. Back it up. In the entire mouse, you have the LOXP sites followed by the LAC-Z gene. In only the regions where FOXA2 is expressed, you also have Cre recombinase. So where you have both Cre recombinase and the LOXP LAC-Z, the Cre recombinase is going to cut out the LOXP sites, which removes the stop codon, and then you will have LAC-Z gene and protein being created. And that's what turns blue. So the blue here is the region where the FOXA2 is expressed, and in the rest of the mouse, you've got the LOXP and the LAC-Z, but there was no Cre recombinase to start transcription and translation of the LAC-Z gene into protein. This is just one example of how Cre recombinase is useful to scientists. There are dozens of other ways that it's useful. For example, what if FOXA2 was involved in a process of development that required a specific other gene to do something first, and we create a mutant in that other gene? Well, by looking at this mouse with the FOXA2 and the Cre recombinase, we would be able to see, okay, well, if that other gene wasn't functioning to do the thing it had to do, how does that change where FOX is expressed, what it's doing, or how does it change the structure of the nodal cord, or whatever other structures that this gene might be expressed in? This here is a part of the neural tube called the floor plate. And the floor plate only exists when the gene Sonic Hedgehog, that we've talked about before, functions. The floor plate is actually created through the activity of Sonic Hedgehog. Well, what if we prevent Sonic Hedgehog from creating the floor plate? What's FOXA2 going to do? Is it gone? Does it just not show up? Does it show up someplace else? These are things we can find out using Cre recombinase. There are dozens of bells and whistles that developmental biologists or just any kind of animal biologist can use to look at the functions of genes with Cre recombinase and LOXP. These are just a few of them. And again, if I teach the developmental biology course next year, I am definitely going to be getting into those. So keep your eyes peeled. And we're getting to the end of the lecture here, so I want to sort of zoom past this. This is yet one more version of the same thing. Another way that that DNA in bacteria can insert itself from a plasmid into a larger chromosome. And in this case, it's called integrons. And integrons are essentially the same thing as recombinases. It's just that they use a different protein, and this protein is called integrase. And the sort of plasmid-like things that get inserted are called cassettes. And instead of using a LOXP site, they use an ATT site. In this, they're called ATTI and ATTC. The ATTC is for the ATT site in the cassette, and the ATTI is for the ATT site in the integron. I can't even tell you how it's different, because it's all the same thing. It's just using homologous recombination spurred by an enzyme to insert DNA through homology. And again, it's reversible. And you can insert multiple sections of DNA into the same spot repetitively because they all match the same sequence. So there you go. Integrons, recombinases, plasmids, these are all just ways that bacterial DNA moves around. This is getting cassettes and plasmids and circular DNAs into other DNAs, and then transposons. If these are inserting themselves into transposons, they get migrated around that way. They can go from a plasmid to the chromosome, from a chromosome to a plasmid, back and forth. And then they can get rolling circle replicated through the F plasmid into a horizontal gene transfer of another cell. It's all just genes and chromosomes moving around in bacteria. That's really what the whole first half of this chapter is about. This shows how they can cut themselves back out again. It's all the same thing. Okay, we have finally finished with that bear. We are almost at the end of the lecture here. I've got like 4 slides left. I might just give you one more. So let's talk about useful mutants, and we have already spoken about one of these before. There are 3 different classes of bacterial mutants that are useful to scientists, basically. We've discussed the antibiotic-resistant mutants, and those are able to grow in the presence of a particular antibiotic. And so what that means is, for scientists, I mean, not the general public, but for scientists, if you have a plasmid that has an antibiotic resistance gene, you can insert the DNA that you're interested in into that plasmid using any of those methods we just discussed, or even others. Most often in a lab, it's done with restriction enzymes. You can put your DNA of interest into the plasmid. You can put the plasmid into a bacterium, which is the next section, and grow the bacteria on media, either a plate or in a liquid culture, that has that antibiotic in it. And presumably, only the bacteria that has your plasmid with your antibiotic resistance gene is able to survive. So it's a great way to grow a huge quantity of bacteria carrying your DNA of interest, and making sure that you don't have other bacteria that don't have your DNA of interest. So antibiotic-resistant mutants are very helpful to scientists that way. There are nutritional mutants, and think back to Beadle and Tatum's arginine mutations in the Neurospora. Now, Neurospora isn't a bacterium, it's a fungus, but the idea holds. Nutritional mutants are bacterium that do not have the ability to synthesize one particular essential nutrient, and they can't grow unless you supply that nutrient. It's the same thing as the Neurospora in Beadle and Tatum's experiments. And then there's carbon source mutants. And by carbon source, we mean sugars. Because bacteria get the carbon that they need to build all of their nucleotides and amino acids and proteins. They get that carbon from sugars. They digest the sugars and hydrocarbons, and they digest those and make larger macromolecules out of them. Sometimes the sugar that they use is lactose, and if they are missing the lactose or lactase gene, they can't grow on lactose because they can't use it. And if they're missing the arabinase gene, they can't grow on the sugar arabinose. The same thing with maltase and sucrase, all of these things. If they can't break down the sugar into smaller pieces that they can use, then they can't grow if that is the only sugar available to them. They will die. And of course, scientists can take advantage of that. Okay, transformation. This is how you get your plasmid into bacteria of interest. And this is what Griffiths was seeing when he was injecting dead S-strain pneumococcus plus living rough strain, or vice versa. At this point, I've already forgotten which is which. When he was injecting the one live and the one dead strain, and it killed the mice anyway, that was DNA transformation. So how does that work? Well, basically, a living cell takes up DNA from outside of itself, and that DNA is most likely from a dead cell. When cells die, their membranes rupture, and their internal parts just sort of float around loose and eventually get degraded or destroyed. But the DNA isn't taken apart one base at a time. It's, it breaks up into fragments, and those fragments break up and denature and break up. and eventually it's taken down to component parts. But when it first breaks up, it's large stretches of DNA or medium stretches of DNA, and those can get taken up by cells. Now, this happens naturally, but scientists in a lab can also do it deliberately. When it occurs naturally, it's very infrequent, but we can make it happen as we wish. One way to do it is through heat shock. You're keeping the bacteria in a lab setting. You usually keep the bacteria on ice and chilled to keep them from replicating and to keep them from dividing. You don't want too many of them. So you have them cold, and then you throw them into a warm water bath, a little bit higher than human body temperature. And that rapid change in heat shocks the cells. And they wind up expanding the pores in their membranes. Bacteria have naturally existing pores in their membranes, and the heat shock makes those pores enlarge in size. And the bacteria winds up ingesting or taking in large quantities of whatever media it is floating in and swelling up in size. And then if you rapidly cool it again, the opposite happens. And when you do that, when it takes in all the liquid that's surrounding it, it takes in, if there's DNA in that liquid and chunks of degrading cell, it takes in all those chunks. And then as it deflates again, when you put it back in the ice, it spits a lot of that out, but frequently retains chunks of it and parts of it. And if some of the parts it retains is the DNA, now you have foreign DNA in your bacterium. So what scientists like to do is they have a test tube full of bacteria, and they put in a bunch of plasmid DNA that they have engineered, and then they heat shock it, and the plasmid DNA gets taken up into the cells and stays there. Or if you don't want to heat shock it, you can do what's called electroporation, and that is you have a test tube, a special type of test tube called a cuvette that has metal plates on it, and then you run an electric current through those metal plates, and that causes an electric current within the media, and that also forces the cells to expand and take in the DNA around them. So that is transformation. So let's look at this. You've got a cell. In this case, they call it donor DNA. Isn't that cute? Because it's like an organ donor. Because it's going to be giving up everything it's got to when it dies. So your donor DNA is broken up once the bacterial host is dead and lysed, and you've got all this DNA floating around in the cell, and it's just kind of doing nothing. And then it gets taken up into the other cell through transformation, and then you get homologous recombination, just like we've been talking about the entire lecture. And if you have different alleles of the same genes in your two different DNAs, then when you get recombination, the alleles are going to trade places. Just like in mitosis and meiosis, like we talked about before. Now, we can do this to map bacterial chromosomes. I mean, sooner or later, you got to realize everything in genetics is about mapping, right? You can map the bacterial chromosomes by seeing what genes transform with each other. So you look at the genotype of your bacteria of interest, and you look at the genotype of the donor DNA, and if they're different, you then transform your surviving bacteria, and you look at its genotype after that. And of course, if the genotypes are about whether or not it has a given resistance gene, or a functional versus non-functional antibiotic resistance gene, then you can see if it has a functional version now by growing it in the presence of that antibiotic. And by looking at the function of the different genes, and whether or not that changes, whether the genotype changes after the transformation, and if it changes in one gene versus two versus three, and which genes they are, you can then figure out what sequence they are in on the bacterial chromosome. So this is the last slide, and we'll stop here. It's hard to see all the pluses and minuses, but in this case, your circular plasmid DNA of the donor is A plus, B plus, and C plus for these three genes. And the acceptor DNA, the plasmid that is circular, or the chromosome that is circular in the surviving bacterium, is A minus, B minus, and C minus. And based on which fragments of DNA get transformed into that surviving bacterium, what you wind up with is different transformants.